Welcome to part 4 of my chapter 6 lecture. We're going to finish modes of the multiplication cycles in animal viruses. We have gotten to the synthesis part, which is where the viral body is replicated. So the synthesis part varies depending on whether you have a DNA virus or an RNA virus. And so DNA viruses are usually replicated and assembled in the nucleus of the host cell. RNA viruses are usually replicated and assembled in the cytoplasm. So that's a key difference. Within RNA viruses, there are positive sense and negative sense RNA strands. And so the positive sense RNA has the message for translation. They can be directly translated into an amino acid sequence, And then negative sense RNA has to be first converted into a positive sense message before it can be translated into an amino acid sequence. Okay, so assembly is just once the synthesis part happens, that the synthesis is making the components of the virus, the protein and the nucleic acid. Then the assembly part is where those pieces are put together. So you have what we'll just call mature virus particles. By virus particles, we just mean viral bodies. Viruses are put together from the pool of nucleic acids and proteins and other components that have been synthesized during the synthesis phase. In general, the first part that is put together is the capsid, the protein coat, and it will be put together as an empty shell that will kind of act like a container or a receptacle for the nucleic acid strand. There have been electron micrographs or pictures taken during this part of the multiplication cycle of animal viruses, cells with masses of viruses that are contained in packets. And one of the important events that leads to or signals the enveloped viruses to be released from a host cell is the insertion of spikes from the virus, viral spikes, into the cell membrane of the host so that the virus can be picked up as it buds off within the envelope. So once the viruses are assembled and they're mature, they can leave the host cell by budding, which is also a form of exocytosis if it's an enveloped virus, or by cell lysis, which just means rupturing for non-enveloped and complex viruses. So in the case of release by budding, the nucleocapsid of the virus binds to the host membrane and it just pinches off um, a little bud and sheds the viruses gradually from the cell. And the cell is not immediately destroyed by budding. Just a little piece of the membrane pinches off to release the viruses. Obviously in cell lysis, it does lead to immediately destroying or killing the cell. The virus is released when the cell ruptures or lyses. So that's a big difference for the two modes of release. Okay, so damage to the host cell is our next topic. That another way of saying that is cyto, meaning cell, pathic, having something to do with pathogenicity, and it just refers to the structural changes in host cells caused by the virus invasion. And so sometimes you can actually view the cell damage under a microscope, and things that you might see would be disorientation of individual cells, major changes in shape or size, and then also intracellular changes, which would be changes inside of a host cell. And those things might be inclusion bodies or syncytium. And inclusion bodies would be something that would be like a compacted mass of viruses or damaged cell organelles in the nucleus or cytoplasm of the host cell. And then the syncytium would be if you have a single cell or cytoplasmic mass that has multiple nuclei, which happens when you have cell damage that causes the fusion of cells. And this table 6.3 from your book has a list of viruses and some of the cytopathic effects in the host cells. The smallpox, causes the cells to kind of round up and causes inclusions in the cytoplasm. Herpes simplex can cause cells to fuse together to form multinucleated syncytia, as well as nuclear inclusions. And there is a picture of the nuclear inclusions in figure 6.15. Adenovirus causes cell clumping and nuclear inclusions. Poliovirus causes cells to lyse but does not have inclusions. Reovirus causes cell enlargement and vacuoles and inclusions to form within the cytoplasm. The influenza virus can cause the cells to round up, but it does not cause inclusions to form. The rabies virus does not change the cell shape, but it does cause cytoplasmic inclusions called negative bodies to form in the cytoplasm. And then finally, the measles virus causes syncytia to form, and you end up with these multinucleated cells, like similar to what happens with the herpes simplex virus. Okay, so with persistent infections, that is going to be when a host cell continues to house or harbor the virus and is not immediately lysed. And this can last weeks or can last throughout the lifetime of the host. And there are some viruses that can periodically reactivate, so become active again. So from a chronic latent state. So you might have periods of time where the virus is inactive or latent, and then periodically when the host's immune system is low or when the host is stressed or there are other things that might cause the host to be more susceptible, that latent virus can become active and cause the host to become sick again. Viruses that can do this are measles, which can remain hidden in brain cells for long periods of time. The herpes simplex virus is one that causes cold sores as well as genital herpes, and those can be latent and flare up periodically. And then also the herpes zoster virus, which is chickenpox, which most people, if they had chickenpox as a kid, they're susceptible to getting shingles as an adult. And what happens is the herpes zoster virus remains latent within the nerve sheath of some of the host cells. And then periodically, if the host is stressed or not taking care of themselves or whatever, susceptible for some other reason, they'll have a flare up, and that's what shingles is. Okay, so damage caused by viruses. Some animal viruses can, when they enter a host cell, permanently alter the genetic material of the whole host cell, which can actually result in cancer. These viruses that can do this are called oncogenic viruses, and the effect that they have on the host cell is referred to as transformation. And transformed cells that are changed in this way have an increased rate of growth, alterations in the chromosomes, and they have the ability to divide for indefinite time periods, which can result in tumors. That's what a tumor is. It's uncontrolled cell division and it can form masses of tissue. Mammal viruses that are capable of causing tumors are called oncoviruses. So the noun is an oncovirus and the adjective is oncogenic. Examples of oncogenic viruses are papillomavirus, which can actually cause cervical cancer, Epstein-Barr virus, which can cause Burkitt's lymphoma. Okay, so now we're going to move on to the multiplication cycle in bacteriophages. A bacteriophage is just a virus that infects bacterial cells. So the most widely studied bacteriophages are the ones that infect E. coli. And the multiplication of bacteriophages has several stages that are similar to the ones that we talked about for animal viruses. And once the nucleic acid penetrates into the cytoplasm, then uncoating is not necessary for bacteriophages. Release of bacteriophages results from cell lysis and is triggered or induced by viral enzymes and an accumulation of viral bodies. And that is called a lytic cycle, lytic referring to lysis. Okay, so the steps in bacteriophage replication are adsorption, penetration, replication, assembly, maturation, and then the last one is lysis and release. Very, very similar to the stages in animal virus replication. So adsorption is the same, it's just the binding of the virus to the molecules on the host cell. Penetration is the same, it's when the genome enters the host cell. Replication, also the same, the components, the nucleic acids and the proteins that make up the virus are synthesized or produced. Assembly is where those components are put together to form a virus or a viral body or phage. The maturation stage is the completion of viral formation. So it's kind of the completion of the assembly stage. That's a little bit different from what we listed in the stages of animal virus replication. Then the final stage, lysis and release, involves the lytic cycle where, the viral infection comes to completion and the cell ruptures or lyses and the viruses are released. Sometimes the virus can cause the host cell to enter a reversible state of lysogeny, and then the viral DNA can be incorporated into the host genome. So that's a reversible state of lysogeny. This figure is kind of hard to see on here, but it is in your book. It's figure 6.16. And it is showing the different steps of the bacteriophage multiplication cycle, starting with adsorption, penetration, duplication, assembly, maturation, and then the lysis and release. So this is showing closer up. This is the adsorption and penetration. And the nucleic acid from a bacteriophage is just injected into the cytoplasm. So it doesn't need to be uncoated. Okay, and then there's a table that has all of the stages listed out for you, table 6.4. So lysogeny is what happens when certain DNA phages called temperate phages go through the process of adsorption and penetration, but don't replicate. The viral genome is inserted into the bacterial genome and then becomes what's called an inactive prophage. The cell is not lysed. Then what happens is that prophage is retained in the bacterial genome, and during cell division of the bacterial cell, then that DNA from the virus is replicated and results in transferring the temperate phage genome to all of the offspring of that host cell. That is called lysogeny. And that results in the spread of the virus without killing the host cell. The genes in the bacteria